Biotechnology. Biotechnology is the manipulation of biological organisms to make products that benefit human beings. It contributes to such diverse areas as food production, waste disposal, mining, and medicine. Modern achievement of biotechnology includes the transfer of a specific gene from one organism to other by means of transgenics, the maintenance and growth of genetically uniform plant and animal cell culture called clones, and the fusing of the different types of cells to produce beneficial medical products such as monoclonal antibiotics, which are designed to attack specific type of foreign substance. The first achievement of biotechnology were in food production, occurring about 5000 BC. Diverse strains of plants or animals were hybridized crossed to produce greater genetic variety. The offspring from this in biotechnology were in food production, occurring about 5000 BC. Diverse strains of plants or animals were hybridized crossed to produce greater genetic variety. The offspring from these crosses were their selectively bred to produce the greatest number of desirable traits. Repeated cycles of selective breeding produce many present day from production programs. In 1960s, an important project by hybridization followed by selective breeding to increase food production of quality of wheat, rice, and crops. Application of biotechnology. Has been applied in various fields. In waste management, it is used to create new biodegradable materials. One such as materials is made from lactic acid produced when the individual lactic acid molecules are joined chemically. Thus, they form a material that has the properties of plastic but is biodegradable. Widespread production of plastic from this material is expected in the future. Biotech is also applied in the mining industry. Bacterium, Bacillus feroxidans can use the molecules of copper to form the compound copper sulfate, which can be treated chemically to obtain pure copper. This mining produce is used only with low-grade ores. As high-grade deposits are exhausted, the amount of copper used in this process will likely increase. In the field of medicine, the most dramatic application in biotech are employed factor VII was produced for use by hemophiliacs. In this biotechnology produced the human gene that codes for the blood clotting protein is transferred to hamster cells grown in tissue culture.
Genetic engineering and recombinant DNA technology are closely related fields within biotechnology that focus on the manipulation of genetic material to achieve specific outcomes. Below, I will explain the techniques involved in these processes and discuss the application of DNA profiling. Genetic engineering definition and techniques. Genetic engineering refers to the direct manipulation of an organism genome to introduce new traits or modify existing ones. This can be achieved through various techniques including gene editing. Techniques like CRISPR to Cas9 allow for precise alteration in DNA sequence, enabling the addition, deletion, or modification of specific genes. Number 2. Gene transfer. This involves introducing foreign DNA into an organism, often using vectors such as plasmids or viruses to carry the desired gene into the host cells. Number 3. Gene cloning. This technique allows for the replication of a specific DNA segment. The DNA of interest is inserted into a vector, which is then introduced into a host organism where it can be replicated and expressed. Number 4. Transgenic organism. These are organisms that have been genetically modified to contain genes from other species, enabling them to express new traits such as pest resistance in crops or insulin production in bacteria. Recombinant DNA technology. Recombinant DNA technology is a subset of genetic engineering that specifically involves recombining DNA different sources to create new genetic combination. The key steps include: number 1, isolation of DNA. The desired gene is extracted from the source organism. Number 2, cutting DNA restriction. Enzymes are used to cut the DNA at specific sites, creating fragments that can be manipulated. Number 3, ligation. The cut DNA fragments are joined together using DNA legacy, forming recombinant DNA. Number 4, transformation. The recombinant DNA is introduced into a host cell where it can replicate and expresses the new genetic material. DNA profiling. DNA profiling, also known as DNA fingerprinting, is a technique used to identify individuals based on their unique DNA characteristics. It involves analyzing specific regions of the DNA that vary greatly among individuals. Here's how it relates to genetic engineering and recombinant DNA technology. Technique used. DNA profiling typically involves PCR or polymerase chain reaction to amplify DNA samples, followed by gel electrophoresis to separate and visualize the DNA fragments. This allows for the comparison of genetic material from different individuals. Application. DNA profiling is widely used in forensic science for criminal investigation, paternity testing, and identifying genetic relations. It can also be applied in medical diagnostics and research to study genetic disease. In summary, genetic engineering and recombinant DNA technology are powerful tools that enable modification and manipulation of genetic material for various applications, including the development of GMOs and pharmaceuticals. DNA profiling, while distinct, utilizes similar techniques to analyze genetic material for identification and forensic purposes.
Gene splicing overview and techniques. Gene splicing is a post-transcriptional modification in which a single gene can code for multiple proteins. Gene splicing is done eukaryotes prior to mRNA translation by differential inclusion or exclusion of regions pre-mRNA. Gene splicing is an important source of protein diversity. During a typical gene splicing event, the pre-mRNA transcribed from one gene can lead to different mature mRNA molecules. Generate splicing enables a single gene to increase its coding isoforms that are structurally and functionally distinct. Gene splicing is observed in high proportion of genes in human cells. About 40 to 60% of the genes are known to exhibit alternative splicing. Gene splicing mechanism. There are several types of common gene splicing events. These are events that can simultaneously occur in the genes after the mRNA is formed from the transcription step of the central dogma of molecular biology. Exon skipping. This is the most common known gene splicing mechanism in which exons are included or excluded from the final gene transcript, leading to extended or shortened mRNA variants. The exons are the coding regions of a gene and are utilized in various cell types for a number of functions. Intron retention. An event in which an intron is retained in the final transcript. In humans, 2 or 5% of the genes have been reported to retain introns. The gene splicing mechanism retains the non-coding portions of the gene, can leads to a deformity in the protein structure and functionally. And the last part is alternative 3 splice site and 5 splice site. Alternative gene splicing includes joining of different 5 and 3 splice site. In this kind of gene splicing, 2 or more are alternative 5 splice site complete for joining to 2 or more alternative 3 splice site.
Food production. Recombinant DNA is used to combat one of the greatest problems in food production, the destruction of crops by plant viruses or insect pests. Science also have developed transgenic potato and strawberry plants that are frost-resistant potatoes, corn, tobacco, and cotton that resist attack by certain insect pests, and soybeans, cotton, corn, and oilseed source of canola oil that have increased resistance to certain weed-killing chemical called herbicides. DNA has also been used to improve crop yield. The transferred gene causes the new plant to produce more grain and a shorter stalk with fewer leaves. The shorter plants also resist damage from wind and rain better than taller varieties. Pollution control. Genetically altered bacteria can be used to decompose many forms of garbage and to break down the petroleum products, and recombinant DNA can be used to monitor the breakdown of pollutants. Medicine. The hormone insulin is grown in large quantities because this is available only in limited quantities from hogs. Another important genetically engineered drug is interferon, a chemical produced by the body in tiny amounts. Engineered interferon is used to fight viral diseases and as an anti-cancer drug. Scientists also employed recombinant DNA to produce medically useful human proteins and animal milks. The egg of animal is injected with the linked genes and will produce the human proteins only in the in their milk. In this way, sheep's milk is used to produce alpha-1 antitrypsin, used in the treatment of emphysema. Cow's milk is used to produce lactoferrin, a protein that combats bacterial infections, and goat's milk is used to produce TPA, a blood clot dissolving enzyme. Moreover, gene splicing is also important in the general process of gene therapy. Finally, recombinant DNA is also used in the production of vaccine against disease. A vaccine contains a portion of an infectious organism that does not cause severe disease but cause the body's immune system to form protective antibodies against the organism. When a person is vaccinated against a viral disease, the production of antibodies actually a reaction to the surface proteins of coat of the virus with recombinant DNA. Altered cowpox is now being used against hepatitis, influenza, or herpes simplex viruses. In humans, recombinant DNA, the basis of gene therapy. Gene therapy is now being used for diseases such as cystic fibrosis, emphysema, muscular dystrophy, and some cancer. Cloning. In the cloning method, scientists remove the DNA containing nucleus from female eggs and replace it with a nucleus from animal of a smaller species. The scientists then place the egg in uterus of a third animal known as the surrogate mother. The result is the birth of an animal that is nearly genetically identical to the animal from which the nucleus was obtained. Such an animal is genetically unrelated to surrogate mother.
GMOs have whispered application. They are used in biological and medicine research, product of pharmaceutical drugs, and experimental medicine and agriculture. Transgenic microbes. In the past, bacteria were the first organism to modified in laboratory. These organisms are now used in producing large amounts of pure human proteins for use in medicine. Genetically modified bacteria are used to produce the protein insulin to treat diabetes. Similar bacteria to produce clotting factor to treat hemophilia and human growth hormone to treat dwarfism. Transgenic bacteria can reduce the formation of cavities. Transgenic microbes can kill or hinder tumor, facilitate crops growth, and produce chemicals which are toxic to crop pests. Transgenic animals. Transgenic animals are used as experimental model to perform tests with genes whose function is unknown. Transgenic fruit flies are used to study the effect of genetic change on development. Transgenic mice to study cellular and tissue species response to disease and that can cannot be studied in mice. Transgenic fish is created to increase the transgenic plants. Transgenic plants are engineered to possess several desirable traits, including resistance to pests, herbicide, or harsh environmental conditions, improve product shelf life, and increase nutritional value.
In conclusion, genetically modified organisms are alive. They have the ability to migrate and proliferate globally. Incidentally, clear signals ought to be delivered to biotechnology laboratories to proceed with added caution and evade instigating unintended damage to public human health, to biodiversity, and the environment. While it is widely assumed that consumers have the rights to demand mandatory labeling of GM food product, self-regulating evaluation of for safety and environmental effects, are accountability for any harm linked with GMOs. Internationally, the leaders must look into these concerns of this technology-driven society. Food and crops that is modified that being used today. Corn. Corn is the most common grown crop in the United States, and the most of it is GMO. Most GMO corn is created to resist insect pests or tolerate herbicides. Corn is a GMO corn that produce proteins that are toxic to certain insects, pests, but not to human, pets, livestock, or other animals. Soybeans. Most GMO soy is used for food for animals, predominantly poultry and livestock, and making soybean oil. It is also used as ingredients like lecithin, emulsifier, and protein in processed food. Potato. Some GMO potatoes were developed to resist insects, pests, and diseases. In addition, some GMO potato varieties have been developed to resist bruising and browning that can occur when potatoes are packed, stored, and transported, or even cut in your kitchen. Canola. GMO canola is used mostly to make cooking oil and margarine. Canola seed meals can also be used in food for animals. Canola oil is used in many packaging foods to improve food consistency. Most GMO canola is resistant to herbicides and helps farmers to more easily control weeds in their field. Summer squash. GMO summer squash is resistant to some viruses. Squash was one of the most first GMO the market, but it is not widely grown.